Hi everyone. Today, I'm gonna go over Squamate, mesotocin, and vasotocin. The background is a brown anole, which is part of one of the papers that I'm going over. It was taken by Baisley in 2018. I got it off of iNaturalist. So, both of these papers are going to be going over mesotocin and vasotocin, which is a mammalian homologue for oxytocin and vasopressin. They basically kind of tag team together, and they regulate social behavior a lot. So I'm sure we've all heard of oxytocin in mammals, and basically these papers are going to examine how they work with different social contexts, and specifically courtship and maternal care. And the image that we're seeing here, I thought I should probably include it because of the different, homologs and stuff like that. It's basically the evolution of oxytocin and how it went into mesotocin, isotocin in fish, and vasopressin and vasotocin and all that. I got it from a paper by Gui and colleagues in 2008. So again, we're talking about the involvement of the oxytocin homolog, mesotocin, and we're going to do it in sexual and aggressive behaviors of the brown anole. It was by Kubelik and Magruder in 2014, and I got it from Biology Letters. They actually have done a couple of good articles on anolis behavior, which is pretty cool. And then the other one is where they blocked the vasotocin receptor in pregnant and postpartum female pygmy rattlesnakes, and Lind and colleagues in 2017 did this, and I got it from Biology Open. And then the image is just a picture of a pygmy rattlesnake, which is the other animal that we're looking at, and it was taken by Scott in 2015. I also got that off iNaturalist. The authors, I thought it was pretty interesting because I didn't realize before, because I've had this paper for a little bit on my phone because I collect papers. One of the authors of the anole paper, he works at Thompson Rivers University right now, but he got his PhD from ASU, and I had no idea about that. And it's like, oh, coincidentally, I'm going to ASU while this dude wrote one of my favorite papers on anoles, and he got his PhD there. So I thought it was pretty cool. And then his picture's on the left. And then he still researches behavioral neuroendocrinology, and he does both avian and non-avian reptiles. So he also researched birds at one point too. And then the other guy, Craig Lind, from the rattlesnake paper, he works at Stockton University, and that's his picture on the right. And he still studies reptiles and ecology and all that. I'm pretty sure from the list of things that I looked at, he exclusively studied reptiles, so I thought that was interesting. The anole study, they used male brown anoles, and then we examined courtship with a female present and aggression with a male present. They basically had the second lizard in a terrarium, and it had like a divider, and then whenever they were going to go study the actual behavior, they took the divider out, so like the animals were able to interact directly with each other. And they exposed the male anoles, which is what we were studying, to social encounters, and then about an hour after, they collected the brains, which, I mean, I know it sucks, but like that's what they did. And they did it in like various baths and stuff like that, and so then we used immunohistochemistry for FOSS to look for like recent neural activity. And then we examined the mesotocin, which again is oxytocin, their equivalent. We looked at mesotocin producing neurons in the paraventricular and the supraoptic nucleus and the thalamus. So that's where it comes from in a lot of animals too, and that's where it comes from in reptiles. So we're looking in the correct place. And a subset of the anoles also co-express a corticotropin releasing factor, which it co-expressed with the mesotocin. And we wanted to look at how it activated in the magnocellular versus parvocellular neurons in the thalamus, because we were trying to find specifically which cells lit up whenever it came to these different social contexts. And again, this is Kubelik and Magruder in 2014 that looked at this. For the rattlesnake study, Lind and colleagues in 2017 looked at female pygmy rattlesnakes after giving birth. So basically, they put these animals in outdoor enclosures, and they put like a divider of sorts, and then they kind of had like this blind so that the researchers could look at them without the snakes actually knowing, so they wouldn't interfere. And we injected a vasotocin receptor antagonist. So basically, we're blocking the VT1A receptor, which is a arginine vasotocin receptor specifically, and we blocked it. So basically, this particular VT1A receptor is homologous to the mammalian V1A because, vasopressin, vasotocin, all that. We looked at a control group without a blockade to see, if we were comparing for body condition to see, if a female is in really bad shape, she might not, dedicate as much time or care to these babies as they would if they were in good shape. And we wanted to see how they aggregated with their offspring. So, did they kind of group up with the offspring and get together with it, and did they touch them a lot? Or, what did they do? Did they coil around them to protect them and stuff like that? So we looked at it up across 4 days after they, gave birth to these snakes. For the annul study, we're looking at FOS activation again. So for the courtship, we saw that there was significant FOS activation, which is, neural activity, in the parvocellular paraventricular mesotocin neurons. So the superoptic one wasn't necessarily too great, and the activation correlated with courtship frequency. So basically, if we courted a lot, then we're going to have more activation than if we just did it, a little bit. And then the courtship also increased the parvocellular mesotocin, the CRF neurons. So basically, the CRF is working along with the mesotocin and the ones that had that activation. For aggression, we didn't really see much going on, so that was kind of just like an insignificant thing, which is why I didn't really mention it to begin with, because, if it was insignificant, then, why mention it? But I guess I probably should have. For the magnocellular ones, they didn't really show anything significant, like I said, or it was kind of weak, so it wasn't really good enough to be considered. And then the interpretation is that the mesotocin was tied specifically to sexual behavior and not aggression. Here we're looking at some findings that we saw in there. So basically this is arranged in columns, and we're looking at kind of zoomed in. So whole brain, and then the part of the brain which is the thalamus, and then we're zooming on specific cells. And these arrows are pointing to specific cells that are activating. So the FOS ones are red, and then the mesotocin ones are going to be green, and the CRF is going to be purple. And we're staining for different things because, like I said, it's immunohistochemistry. And then we're also looking at the social behavior and the neuronal measures. So, like I said, it was a frequency of social, courtship behavior specifically. And we're going to look at the FOS activation. So the x-axis is going to be the social behavior, and then the y-axis is going to be the activation, which we're seeing the trend going up. For the rattlesnake study, like I said, we blocked the VT1A receptors, so we're blocking the vasotocin, and it disrupted the maternal aggregation. So they weren't necessarily clumping together with their offspring as much, and they weren't really touching them as much. They didn't spend as much time with them because basically the females went to the other side of the divider that they had in this enclosure, and they wanted to spend more time on the other side away from their offspring than it was, with their offspring. We're looking at maternal behavior and controls, so they maintained their behavior and the vasotocin, without the vasotocin being blocked. So they did protect their young, and they did aggregate with them, and they did want to touch them more. So basically, we discovered, or Lind and colleagues discovered, that the vasotocin is critical for maintaining this care. that we're seeing with, aggressive snakes postpartum and stuff like that. And if they don't have it, then they just don't really care about investing in their offspring. I thought that this figure from Linden Collies was pretty good because it kind of like directly explained what happened postpartum. So we're looking at the fact that the controls are spending more time with their offspring, and the ones that got the blockade are definitely spending a bit less time. And we're looking at, for this one, we determined that mesotocin is definitely for courtship and sexual motivation, but not as much for aggression. And then vasotocin is for maternal behavior, because like I said, they kind of tag team and everything. So, because they're being found around the same spot. So, and then in mammals, we're seeing them tag teaming together and all that. And then it kind of determines the same thing in reptiles. So it does mirror the mammalian roles of oxytocin, vasopressin, so like bonding, parenting, reproduction. And actually, the rattlesnake paper by Lind mentioned the fact that these antagonists have been proven in reptiles and mammals. So, like I said, it suggests similar roles. And the mesotocin has previously been examined in turtle nesting behavior. So again, that kind of proves itself there too. And it regulates courtship behaviors in mammals and birds. And so, we kind of conserved these systems evolutionary. And so I think that that's pretty cool, because people tend to think that reptiles, just are kind of instinctual only. And it's like, we have these systems where people just kind of associate oxytocin with love or whatever. And then we have, these maternal behaviors, but they're being preserved in reptiles too, which are, not super well understood. So we have established that the parvocellular PDN neurons are activated during courtship, but not the magnocellular ones. And we've also established that the vasotocin action is mediated during specifically the VT1A receptors and not just like a hormone dump. In anoles, the courtship activates parvocellular neurons. In the rattlesnakes, we have the VT1A receptor blocked, so then the mothers don't want to aggregate or protect their offspring. So it shows a context-specific role in social behavior, because, with the anoles, it didn't really activate during aggression, but, it did during courtship and activated even more during courtship. And it demonstrates, the evolutionarily important role of these two in social behavior. And like I said, like we saw at the beginning, it turned into vasotocin or vasopressin and all that with the evolutionary tree that we saw. And then there's all my references, including the images. So I hope you guys thought it was interesting. I know it was filled with jargon, but I think it's cool, and these are some of my favorite papers about that. So I hope you guys have fun, and maybe I'll see you guys again.